Thank you for the intro, and thanks for having me and coming here today. I'm going to talk about this project at the Diagnostic Development Program about identification of the prognostic gene signature in this clinical trial study of early ER-positive breast cancer patients. Breast cancer is the most commonly diagnosed cancer in Canada, and in the Canadian Cancer Statistics dataset, the projected new cancer cases for females, 25% of all cancers are diagnosed as breast cancer, and then also the majority of female breast cancers are going to be diagnosed at an early stage. So this brings up the importance of prognostic and diagnostic approaches that we take for these female patients. The ER-positive or luminal breast cancer are very common type, the most common actually type of breast cancer. Over 83% of newly diagnosed patients are among ER-positive, HER2-negative, or ER-positive and HER2-positive cancer types. And also the death rate by this subtype is very high among the patients. There's a very good successful story of Herceptin and also HER2 targeted therapies for HER2-positive patients that you know about Herceptin or lapatinib. They're all targeting HER2-positive tumors, but there's limited improvement in targeting ER-positive cancers. These two data sets are coming from large population studies in UK and US. Unfortunately, majority of world population is not considered in these reports, but it's a representative of breast cancer population. So the first treatment for these types of patients is endocrine therapy or anti-hormonal therapies. For example, I just bringing two drugs, Tamoxifen and Examostane, because this clinical trial was focused on these two drugs. Tamoxifen is a selective estrogen receptor modulator. It affects by stopping the estrogen receptor activity in the cells. Estrogen receptor is present in many tissue types, and it affects the cells by promoting cell proliferation. So these tumors that are highly dependent on the presence of estrogen receptors, so they highly express estrogen receptors, so they are highly dependent on the presence of estrogen hormone. By blocking that, we will affect the tumor's proliferation. Examostane is an estroidyl aromatase activator, which inactivates the activity of this aromatase enzymes by stopping the source of estrogen, because these enzymes are involved in conversion of androgen to estrogen in peripheral tissues. So these being effectively treating hormone-positive breast cancers, but there's still a high risk of recurrence, and especially in a long term, which these patients are at least for 20 years under the risk of relapse or metastasis. So by this stats saying like about 20 to 50% of all women that being treated could still have the chance of relapse and metastasis in 20 years. So we need to come up with methods to stratify cancer treatment. Some of the breast cancer patients might not need much of treatment or therapies because of that. These treatments have very high side effects, have very severe side effects. So if they don't respond well, then we shouldn't affect their life quality. And then we should decide if the anti-hormonal therapies are enough for these patients, or define the patient groups that might benefit from alternative therapeutics. So the aim of this study is to identify this gene signature that can predict the risk of recurrence in this population of breast cancer. So we used the team clinical trial study. That's the Tamoxifen and Examostane Adjuvant Multinational trial. It's being, all these countries being involved in this clinical trial of early ER+ and progesterone positive breast cancers that have completed primary therapy, and they're most menopausal patients. So with this taking into mind that we need to consider all these clinical factors to reduce the biological variance of the patients, and we selected a 350 patient cohort to come up with this gene signature. So an overview of the steps that I've taken is we started with that cohort of 350 patients, selected a NanoString gene panel to do the expression profiling of patients, and after some statistical analysis and taking into consideration a functional approach of pathway gene sets and considering the clinical and survival information of the patients, I applied the Sims method to calculate gene coefficients and selected the signature, and I will go through in detail to explain the methods. So starting with the gene expression profiling, NanoString is a powerful method when we have low quality samples. For example, in this case, we don't have a high tissue amount of these patients, and they've been preserved over many years. So it gives us that power, and we selected the PanCancer pathway panel, which is focused on cancer-related pathway targets. It included about 800 genes that mostly came from these 13 pathways, and additional some extra genes that could give us more information. So then this is the method that being developed at OICR by Boutros Lab. Subnetwork integration of multimodal signatures, or SIMS as in the short term, is using the for example, it uses molecular profiles. For example, in this case, we had our NanoString PanCancer panel of mRNA levels and clinical information that is used in this model, as well as our pathway gene sets, and also as well as the genes' functional interactions, as considering them as each pathway into a subnetwork of interactions. Then using the Cox proportional hazards model, there's coefficients calculated for each gene and also the interaction. So then there will be a number of subnetworks or single subnetwork of a pathway selected to calculate the risk score for each patient in the model. And that will give us the power to categorize our patients into the different risk groups by calculating the hazard ratios between them. So to bring an example and go to the details of the model. This is the formula for a Cox proportional hazard model. Each gene's coefficient is calculated as this beta in the model. To make it simple, a hazard function is just trying to relate the time to an event that occurred with the biological variables. In this case, our X would be the expression of the gene. It can be considered as a continuous variable or grouped by just high and low expressor groups of samples. And this beta, which is also defined as the hazard ratio in the different format, is what the model comes up with. So then we calculate also the coefficient of the edge. So consider a network as this. It consists of nodes and these edges that are showing interactions between the pair of genes in our example. Each node is a gene. Each edge is defined by the interaction between the two genes. And this is how we calculate the coefficient for an edge. We go with a multivariate Cox proportional hazard fit, where we have the first gene involved in the pair interaction, the second gene, and this will be our coefficient for the edge, where the expression, the complement of exclusive disjunction of the two gene groups, as if they were both high expressors or both grouped as low expressors into the two genes, will be considered as the group, one group, and the rest of them will be the other group of samples. So then, bringing an example, this is the apoptosis pathway where we have used the gene sets from Reactome and functional interactions of the functional interaction network database of Reactome to make these network of pathways. In the apoptosis pathway, we have 85 genes as our nodes, and then there are over 400 edges as gene pair interactions, but we only consider the ones that are significant with a threshold, so we don't take into account all the genes that might not group our patients into two distinct significant groups. So in this case, 13 genes and 14 interactions were accepted after our cutoff. So from here, we did a kind of repetitive approach. We took these gene sets for each pathway, calculated the risk score, and evaluated the performance of the model using that single pathway gene set. Then selected and combined the best performing gene sets in a repetitive way. Again, if some of them were better, then we repeated the method. So the patient's risk score for each of those pathways is calculated in two ways again. The genes only model is taking that hazard ratio for the single gene times the expression value for that patient, and the sum of all the genes involved in that pathway gene set makes the risk score for that patient sample. And then the interactions model is taking in both genes' expression levels. It's, in this case, also as the Sims method concluded, the genes-only model could be a better predictor of patient's outcome. We also evaluated the interactions model and ended up using only the gene sets. So the results, these are the top two pathways that were validated significantly. The apoptosis and cytokine-cytokine interaction receptor pathway, including 13 and 10 genes, were best performing in predicting the risk in the patients with the hazard ratios over 2.5. And then combining these two gave us a better predictor. We tried combining different pairs and more than 2 pathway gene sets in the model, and also found that these 2 pathway gene sets have a lot of functional interactions between them, and 4 of them are overlapping. Unfortunately, because this study is not published yet, I cannot disclose the gene names, but yeah, this is how they interact with the 19 genes. There are 4 common between both gene sets, and they have, according to Reactome Functional Interaction Network, they have very close functional interactions together. Looking further into these 19 gene sets, 19 genes, this is a distribution of the expression in our study. So you can see that some of the genes expression were higher in the low risk patient group, and some are the opposite, and that shows that's also considered by the coefficient calculated for each of them. Some of them have a positive or negative coefficient value. Then we should consider if the clinical characteristics and the gene signature will stay prognostic after considering all the clinical features of the data. And this is how the gene signature performs in a multivariate model with a hazard ratio over 4. So then we further evaluated this signature in an independent expression data set, which I used MetaBreak study which is available at the CBI portal for cancer genomics. The only thing to consider here was that the platform here is different. It was a Illumina bead array expression analysis where we had NanoString data. This was taken into account by just scaling all of the expression values to reduce the effect of the different platforms. And the disease-associated overall survival in 10 years were considered in only selecting the cohort of ER-positive and postmenopausal patients. So this gave us a group of over 1,000 patients, and the overall survival in 10 years still could be predicted by the gene signature. So in summary, our prognostic signature, including these 19 genes, demonstrated the prediction of the risk of recurrence in ER-positive breast cancer patients, and the pathway gene sets provided the functional relevance to our prediction model, but the gene interaction model could not perform as well as the genes only. This could be suggesting that the gene mRNA expression level only can define the difference between the patients, or I'm interested in looking into changing that method of taking the interactions into the model and further analyze that. These genes were among apoptosis and cytokine receptor interaction pathways that suggests that they have a critical role in relapsed patient population, and this signature also remains prognostic. We can further use this to predict better alternative therapies for these patients that are at a high risk of relapse, and as well as using other predictors of the risk in the patients. These are the references that I used, and thanks for our OICR team and also the UK Clinical Trials Unit.